Hi guys, welcome back to the third and final lecture in the series on classical genetics. And as I said in the last uh, video, um, dominant recessive is the way we introduce the topic. But the truth is, traits are rarely inherited in this either or kind of situation. You're not smart or dumb. You're not tall or short. You have lots and lots of different possibilities. Your eyes aren't brown or blue. Um, and so, while Mendel gave us a good start, what we found is there's all kinds of other ways in which traits are expressed, right? Alternative modes of expression. Um, think about why I use this photograph. So remember, one of the ways I want you to think about this is in simple terms. Dominant recessive is where you either are one or another. It's an either or kind of situation. There's another way for us to express traits, and that's called codominance. In codominance, we see both traits expressed at the same time. Then there's incomplete dominance, and these two are easy to mix up. So sometimes I'll say, instead of incomplete, I'll say pink complete. Because in incomplete dominance, we can get a blend of traits in the heterozygote. So what do I mean? All right, look at these two photographs. Which organism shows incomplete dominance and which organism shows codominance? I'll give you a second. It's the zebra that shows codominance. Look, both traits are expressed at the same time. It's black and white at the same time. The wolf up top there is actually showing incomplete dominance, where it's a blend of traits, right? It's kind of a gray wolf. It's a blend, or it's a brown, tawny brown. It's a blend of brown and black. I don't know what you call it, but it's a blend. Got it? Now, there are traits also that simply aren't the result of just two alleles. So imagine this. Imagine if I can have a brown eye allele, then I can have a blue eye allele. Couldn't I have a green eye allele as well? Another copy of the gene that's different from brown and blue? Of course, the answer is yes. The classic example we use in high school biology for multiple allelic traits is the ABO blood group, and we'll talk about that in a second. There are sex-linked traits, that is, traits that are most often that are connected to the sex chromosomes. Remember, the sex chromosomes are X and Y. Y is this little piece of crap kind of a chromosome, not much on there. I think about 13, 15 genes, maybe 20. I don't know. But the X chromosome is a big chromosome. I think there's 3,000 different genes there, and diseases that are caused by mutations on the X chromosome are called sex-linked diseases. X chromosome. Last but not least are the polygenic traits, and the truth is that most traits are polygenic. Your height isn't determined by one gene, and you're not going to be tall or short. You'll have lots of different possibilities. So polygenic traits are traits that are the result of interactions between multiple genes. Let's talk about incomplete dominance. Remember, I called it pink complete dominance. Let's take this true breeding red flower and a true breeding white flower, right? So they only have the big R gene for red, or little r for the white flower. When I breed them together, what do I get? Well, I get all heterozygotes. But instead of having all red outcomes, what I'll have is a blend. Remember, pink complete is an example in which traits can be blended. So all the heterozygotes will be pink. Straightforward enough. On the other hand, I could have codominance, right? Imagine I have a true breeding white bunny and a true breeding black bunny. I breed them together. I get the heterozygote. And what's it look like? Well, its phenotype is going to be both at the same time. Look, it's a black and a white bunny. Maybe it could have spots, it has stripes, whatever, but that's the example. In codominance, you see both phenotypes at the same time. And here's the Punnett square to show you how that can happen as well. Huh. It must be nice living on an island full of bunnies. Next is our example of multiple alleles, and not to make it too complicated, but you also see codominance in the ABO blood group. Anyway, multiple alleles, as I said before. Imagine that I can have not just one eye color gene or the second eye color gene, blue or brown, but imagine a third eye color gene. Well, that's what we have in the ABO blood group, and there's really three possible genes. There's IA, IB, and then little I. Well, what does the I stand for? Well, it refers to a group of proteins in our blood called immunoglobulins. They're part of the immune response. They're called antibodies. Anyway, back in the day, people found out that you couldn't really mix anybody's blood together without typing it because people have antibodies. And so if I'm type A and you're type B and you give me your blood, my body will attack your blood because it's not type A. It's different. And so these differences are measured with immunoglobulins. But anyway, they allow us to create these phenotypes based on this multiple allelic model. So you could be type A. You could be type A. If you have both copies, then you could be type AB. That's where this is codominant as well. And then last but not least, if you have two little i's, then you're called type O. That's a pretty rare condition. And you know what? This also shows this dominant recessive kind of thing happening as well. Type A blood can also be the combination of IA and the little I, where little I is recessive, and type B blood can be the result of IB with an I as well. So these are fun problems to do. They actually require you to do a little brain work. And so we give an example here. I've got the homozygous type male B. So here's his genotype: big I big B, I big B, and he's going to breed with a heterozygous female who's type A, but she's a heterozygote, so she's IA little I. What's the possible outcomes? Well, I'm going to get kids who are either AB or kids that are type B. You know, 1-to-1 proportion or a 1/2 likelihood. 50% of the kids will be type B. 50 will be AB. What if I take a male that's type O, little I, mate him with a female who's AB? What do I get? Well, I get half type A's, half type B's. Next one's a little more complicated. If a boy has type O blood and a sister has AB, what are the genotypes of the parents? All right. So I know I have to have this situation. I know I have to have this IA-IB. So how do I make a Punnett square that'll allow that to happen? Well, it's got to look like this. All right. So dad is going to be a type IA little I, which allows me to make the AB daughter. And then here's the opportunity to have a type I son. All right, so there's the parental phenotypes. Hard for me to explain how to do this. You got to use your brain. I think you can do it. Remember, we also talked about sex-linked traits. So sex-linked traits are traits that are located on the X chromosome. Remember, the X chromosome, if you're a girl, you have 2 X's, and if you're a boy, you have an X and a Y. Question is this: who shows X-linked traits or sex-linked traits more, boys or girls? And you might think, well, it's an X chromosome, so it must be girls. Wrong. It's the boys. It's tough to be a boy just because if you're a boy and there's a mutation on the X chromosome, you're going to show that disease. An example of sex-linked trait is eye color in fruit flies. Most fruit flies have red eyes, but you have this white eye mutation. One of the things about doing Punnett squares for sex-linked traits is you have to talk about gender plus the trait. And so we use this superscript, just like we saw in the last example of the multiple allele traits, right? So here's dad. He's red-eyed, and he mates with a white-eyed female. What are we going to get? Well, first of all, when this X meets this X, I get a girl. And she's going to have big R, little r, so she'll have red eyes. Here, I'm going to have a boy, but he is going to have white eyes. In fact, I'm going to get white-eyed males and red-eyed females in a 50-50 proportion. Another example of sex-linked traits is colorblindness. It's an X-linked trait. Who shows it more? Boys. So I have a normal male. He's X big BY, and I've got the carrier female, right? She's normal because she's got a good copy of the gene, or her phenotype is normal, but she's secretly carrying this colorblind gene. When I mate these two together, I don't know why this has a little R here. It should be a little B, but what do I get? Well, I'm going to get normal female, homozygous dominant, carrier female, a normal male, and then a colorblind male. So the males have a 25%, or if you're a male, you're going to have a 50-50 chance of being colorblind. Well, these problems are kind of fun. You really have to be specific. A lot of times you'll use language like the male is a homozygous dominant. You have to know what that means, but I think you can sluice it out. I like doing the practice problems. Hopefully you do too. I think the biggest fun, or the most fun problem is the pedigree problems. So, and this is the final part of your material. So pedigree charts are charts to show the inheritance of traits within a family. And the pattern of the chart indicates its mode of inheritance. Is it a dominant recessive gene? Is it X-linked, etc.? And so, what do you need to know? Well, in pedigree charts, circles mean girls, squares mean boys. If it's colored in, that person shows the disease. If it's half colored in, they're the carrier. They're the heterozygote. Okay, so and lots of times pedigree charts may not show that. You'll have to deduce that. Anyway, when you have Bill and Ann get married, they have Ann's a carrier, Bill is a carrier. They have 3 children, normal female. Barbie's a carrier female. Another couple, Karen and Dan. Dan's a carrier in this case. They have 3 kids as well. Normal son, normal daughter, but then a son, Ken, who is a carrier. And when Ken and Barbie get together, they make a daughter who's got the disease, Sally. So, autosomal recessive pedigree chart shows a sort of a dominant recessive trait that's carried on your autosomes, or not the sex chromosomes. It's not sex linked. Here I've got the female. She's got the recessive type, so she's got the disease. And the question is, well, what's dad got to be? And so right away you should start to say, like, well, if I ever see a diseased kid, dad must be the heterozygote. Of course, they have 2 children, both of which are heterozygotes, right? And so that doesn't really tell you anything about dad's characteristics. This daughter gets married to this man. I don't know what his traits are, and they give rise to more heterozygotes, normal appearance. Now this chart doesn't show that half thing, but it's given it to you using the genotypes. And of course, when this female gets together with this male, then suddenly this autosomal recessive disease emerges. They have 2 kids, a daughter with the disease and a son with the disease, but a normal female, but we don't know what her phenotype is or her genotype is either. Okay? Well, that's getting to the end there. Remember, we looked at these different ways for traits to be inherited, and we learned a little bit about pedigree charts. You can see, you can work through those practice problems in your book. As it stands, we're right in the midst of the coronavirus. So if you have any questions, I guess we're going to probably have an online class, and I'll keep you guys abreast of that as we move forward. But one good thing is true, that's it. Summer's coming. Summer's coming.